how to create or creating analysis pipelines. So a usual image analysis pipeline, I think, is divided into three kind of key steps. The pre-processing, or like massaging your data, so doing some filters, doing background subtraction, that kind of stuff. Then segmenting or finding the objects, and then measuring them, or doing some analysis on them. So we'll step through these three steps, or these three kind of bits, and explain them in a bit of detail. So filtering your data. So if you go to the process menu, filter, there's a whole bunch in there. I'm not going to explain every one, but generally these are doing like a blur or some modification on your image. So it's kind of smoothing the data. The way it works is it'll ask you how many pixels. So if you're doing a mean filter, it'll say 1 pixel, 2 pixels, 5 pixels, however many. And what it's doing is it's saying, like, if you have a 1 pixel array, you're going 1 away from each pixel. If you had 2, then there'd be like another row out here and you'd have 25 pixels. And all it's doing is it's saying, well, what's the mean of all those numbers that make that up? So you can do mean, you can do median, they're different ones, but these are the two that you probably use. You might use Gaussian blur as well. But basically, one key difference that I like to point out is if you do a mean versus a median, the median filter will preserve edges a bit better. So depending on what you're trying to do, you might want to be making a, you still want to get like a nice threshold or edge detection of your objects. So running a median might be a bit better for your workflow. The next thing is you might want to do a background subtraction. So it can help remove uneven illumination. So there's this example image from the ImageJ toolkit. And I think it's kind of like a CFU type assay or something. So there's these dots. And if you draw a line and measure it, you can see that the background's kind of lighter on this side as it goes to that side. And when we draw that line, you can see that the background's changing. So if you just did like a mean or median filter that was the rough size of these dots, you'll end up losing some of the dots, and it's not really accurate the way to do it. So you're kind of going to screw up your data if you just do a big filter. So you do a rolling ball background subtraction. And the way it works is if we took that line scan, that's like, you can see the angle. And if we did it over the whole image, you'd see that those spots or colonies or whatever are like little spikes sitting up. And that's kind of useless. But if we inverted the image, then they would be like these holes, right? And so the top flat bit here, which isn't flat, it kind of slants down, is your background. And so if we were to get a ball and roll it across the surface, hence where the name rolling ball comes, we can measure the change in background. And so we just have to make the ball big enough that it doesn't fall in the hole, right? That shows that. So if we roll the ball across the surface, now the ball here is so big that it doesn't fall down. So with these changes don't really affect the center of mass of where that ball would be. And so we can get a bit of an indication of what our general background's doing, normalize the image against that, and then it's all nice and flat. So that's how a rolling ball background subtraction works. So the key thing is, so maybe I'll flip it. It's like, who's done a background subtraction in FIJI? 1 or 2 people. Who just left it at 50 or kind of went maybe 100? You just guessed. You didn't actually scientifically determine what number that should be. That's what most people in my experience do. They just go, oh, 50 looks good, that'll do. Or 10 or 5, whatever. So what you should do is measure the size of these objects and then set that as your radius, right? So that's what we're all going to do. There's this image and it's like some crystals, and you can clearly see that the background's a bit rubbish, right? So it's like a curve. And we have to measure across the smaller axis and set our ball radius bigger than that. So you draw the line over them. I just realized that I haven't shown you how to do measure yet. So you just hit M and it comes up a table with the number. So you can draw a line over a couple of these of that short axis and figure out that they're all about 5 to 10 or 50 to 100. I don't actually remember. And then set your ball radius to be slightly bigger than that. And that would hopefully then give you something like this. Or a nice flat one, which I've just realized I haven't shown you how to do that either. So that's Command K or plot profile. Yes. We'll find out. Yep. So mine are all about 8. So if I go on here, roughly something like that. In the interest of time, I might get us kind of moving on because this is only one part of the workflow. But I wanted to I thought maybe I should highlight why we're doing this. So if we looked at the image in the middle, this one's the raw image, right? So let's assume that we want to measure the area of all of these things. So you could trace around them all. That would be really annoying. So how do we do it? Or how do we select these objects really quickly? And so one way is to do a threshold, which I'm going to cover in the next slide. So I'm not jumping ahead too much. But you can see that it's kind of hard to pick all of them because I can get a pretty good selection at the top here. But once I start trying to get the bottom ones, I start getting all this rubbish at the top, right? And that's because the background intensity is shifting. So once we've cleaned it up with that background subtraction, now it does a pretty good job. So that's kind of the next stage would be to turn all those objects into something that we can find and then measure the area or whatever. So that's what we're going to move on to. So I guess that I just thought I'd give a bit of a lead into the point of this. So this is like I showed you the median filtering, showed you this. There's many different things you can do to kind of smooth the image to get a good threshold or selection. And that's there's one extra slide, sorry. You can do math. So you can do like add or subtract or different math things. And that can be quite useful to make a quick mask or something. So the next step in the workflow from pre-processing, now you want to segment out the objects. So I've got some nuclei. We want to find all these kind of objects or whatever. And that's what segmentation is. So we can manually do it. We could manually trace around things, do like a manual threshold where you adjust it to say this looks good. But ideally, you want to use like automatic algorithms so that it's reproducible, because you moving a slider until it looks right is not reproducible. It usually requires good signal to noise. What I mean by that is that your object that you're interested in should be really bright relative to the background. And you want to make sure that you've got the right resolution and image and canvas sizes and things like that. But what thresholding is it's using the intensity values to divide your image into like foreground and background, or your interested thing and not interested thing. So it's just one cutoff. And the position of where that cutoff is will select which ones to keep, right? So if we look at our histogram of this image, well, there's a bunch of gray pixels, that's these ones. And there's a bunch of blackish pixels, that's these ones. And so how do you draw a line to say, I want to keep the nuclei, but avoid the background? So there's this tool, which is under Image Adjust Threshold. And there's these sliders, and you can move them freely. But as I said, it's not really reproducible. Ideally, it should be done on like your single color images. There is a thing called color thresholding. That's a completely different tool. The output of a threshold is a binary image. So the things that you wanted to keep are set to 255. The things that you say are background are set to 0 or black. And just as I say, be careful, because once you do it, it's not really like an undo thing. So this is where that doing duplicate, and in case you stuff it up, you have something to go back to. So filtering can certainly help. So without a filter, to us, it's still pretty obvious, but to the computer, it might be a bit not as obvious. So running like here, I've used a Gaussian filter. It just makes these even more obvious to the algorithm, where it'll then pick up the 2 nuclei with, this default algorithm. So to show you here, I've just put a gradient, and if I go threshold, you can see that my selection that I'm picking and the gradient is perfectly matching where I'm sliding the slider to. It wouldn't be like that if there was the nuclei. There would be the 2 humps like we saw before. FIJI has a bunch of algorithms. There's this where it says default here. We can change that to all these different ones. They're all like from papers in the 60s and 70s of like signal noise processing of like radio traces and stuff. But basically, there's a tool called image adjust auto threshold, and it just runs all of them. And you can appreciate that, okay, these 2 nuclei are pretty obvious where they are, but there's a few that are more generous. There's a few that are only picking up the little nucleoli. So they're just picking up different decision points where to cut between foreground and background. Once you have a binary image, under processed binary, there's a whole bunch of tools. And so there's the lovely ImageJ diagram down in the corner here, where you can see that it's like a gradient. And then when you threshold it, it's all the same. It's all 1. So here it's black, so it's inverted. But some of these tools, like you can erode the edges, so you can shrink 1 pixel in, so you can eat in from the edge. You can dilate it, well, that makes it kind of fatter by 1 pixel. You can open holes or close holes, and they just kind of run and dilate, then an erode, or an erode, then a dilate. You can skeletonize. You can get the outline. You can fill in holes. You can do all these things. So one useful thing is watershedding, which I'll explain on the next slide. So watershedding is an algorithm where, say, let's pretend these blobs are nuclei. We'll use our imagination. But if we wanted to cut ones that we know are probably 2 objects, but they're joined together based on our imaging. So when we threshold it, it looks like this. So we use this watershedding, which is similar to like watersheds on the side of a hill. And so we do these expansions and dilations to see when we fill up the different like well points and find the midpoint, and then it can cut them, right? So that's a useful tool if you're looking at, say, nuclei and you just want to separate them all. Now, there is like AI tool or machine learning and deep learning tools now to find nuclei. That's really, really good. So, but you can still use watersheding to cut stuff. It doesn't tend to work very well if it's like a immune cell where it's all funky, because it's going to chop it up into like 100 different blobs, and then that's kind of useless. So be careful and make sure if you do run a watershed to look at the output and see if it makes sense. But there aren't any like parameters you can play with for that watershed. So we're going to work through a kind of what I think would be like a somewhat typical workflow. And I'm going to use a cell here. It could a nucleus. It could be a bacteria. It could be a crystal. It could be a bead, whatever. But use your imagination. But we're going to find an object, and we're going to do some things so that we can then measure it. As I said before, we can manually trace around it, add it as ROIs, and split them out. But we'd rather automatically do it. So if we can take our image and then threshold it into something like this, that's the first step. And then we can add it to this thing called an ROI manager, which is quite useful. So if you draw any annotations like squares or circles and that, you can store them here. So if you hit Command T or Control T, they all kind of add. And you can hold them and store them. You can output them so that you can save it for later. So say you've traced around a cell, or you've had cell pose or some algorithm to find your really obscure selection, you can store it so then you can reopen it later. You don't have to redo it every time. And then you can also do things like measure it, rename it, do funky stuff. You can update selections, you can delete them. These buttons are all pretty self-explanatory. But if you had like 50 nuclei here, you could just hit measure and then it measures all of them, puts it in one table, which is really useful. So you can add stuff manually, but you can also use the Analyze Particles tool to add it to the ROI manager automatically. So this Analyze Particles tool pops up this window, and it lets you set up parameters for finding objects. Relatively self-explanatory. Size, so you can, if you know the rough area of your object, you can put in, you know, how many. If there's no scale, it's based on pixels, but if there is scale, it will be based on microns or whatever. Circularity is how round something is. So if it's really round, it'll be 1. If it's not really round, it'll be 0. Most likely somewhere in between. So say you had a mixed population of bacteria and there are rods and cocci, you could just find the really round ones or find the ones that aren't and segment that way. Show, you can drop down and it will be like a mask or an overlay. Again, these are pretty self-explanatory, but if you do nothing, it'll just won't make a new window. And then these ones down here, I say they're pretty self-explanatory. So display results. So if you just want to measure it at the same time, it'll do that. Clear results. So if you already had stuff in the results window, like we were measuring those crystals before, maybe you want to keep things tidy, you can leave clear results. It'll get rid of it first. Summarize will give you a new table that says like, I found 5 objects and the average size was X. Add to manager, we'll dump it into the ROI manager. Exclude on edges. So say you had like half a cell over here. You don't really want to quantify that kind of stuff because you don't know what the other half of the cell is doing. So you can just tell it not to take anything that's touching the edge. To include holes or not. So if it was like a donut, do you want to just have a circle or do you want like an actual with a hole in the middle ROI? I've never used those two and I never remember what they actually do. So not important. One other way that you can kind of detect objects just quickly is using the find maxima, process find maxima. So what that does is you have this noise tolerance, pops up this window. It's kind of fitting like a Gaussian curve. So if you say like 1,000, it's going to find something that's more than 1,000 in brightness difference to its objects around it. So it can be somewhat useful, and you can output like a single point or like center of mass, or you can output things like the segmentation, which will be like a line between the minimas. You can just say count objects. So if your things are really obvious as like just particles or something, you could just use this and it will spit out to the table, I found 50 objects, analysis done, you know. Again, you can use preview and it will show you. So it can be useful. As I mentioned, there's these other tools now. So Stardist is a published deep learning model. If you use it, it does have a plugin in FIJI. Make sure you cite the paper. But it does something like this, where you can have like a whole tissue and it finds all the nuclei. It's really good. So we're going to do it the old-fashioned way in the workflow, but this is something that exists. Likewise, CellPose is a really good deep learning model for finding cells. And it can find really odd-shaped cells. It can find histology stuff. There is now a plugin that links it into FIJI. I've been pulling my hair out for the last week trying to get it working on multi-user computers. But if you want to try it on your own laptop, let me know and I can walk you through it. But that exists. So we're going to do it the old-fashioned way. So what can be measured? We can measure things like descriptor information. So how bright something is. You know, and we might say, our cell had a maximum intensity of this. We can count things, so how many objects there are, how many bacteria, endosomes, crystals, grains of sand, rocks, things like that. How long or how big or how round the object is, how elliptical, they're all descripting things. We can do changes, so you could do FRAP. So if you want to say like, my thing goes to the membrane at this rate, and you use the microscope to bleach away part of it and watch the intensity comes back, we can quantify that change over time. If your cells or whatever are eating things or drinking stuff, or you're injecting stuff into a tail vein and looking at it diffuse through tissue, you can measure that across time and things. And you can do ratio stuff where you do fret and other kind of calculations. So there's lots of stuff you can measure, because really an image in ImageJ is just like an Excel spreadsheet with numbers for each pixel. So by default, you don't get a lot of things turned on to measure in ImageJ. So if you go analyze set measurements, this thing pops up. You can turn on all these lovely different toggles. So some of them are pretty obvious. What's the minimum, maximum value? What's the center of mass? If it draws a bounding box, what's the coordinates of that bounding box? Shape descriptors, so that's where it'll tell you how round an object is, what its perimeter is, or things like that. Integrated density will tell you the sum intensity of the area that you've drawn around. Ferrite diameter, that's the longest possible straight line within the object. So that can be quite useful. You can also do like the display label down the bottom, so that then tells you in your results table that that's from this window and this channel and this Z slice and all that. So that's quite useful to turn on. So here, you've seen this blob image before. I've thresholded it, and then if we wanted to look at its shape descriptors, so you get circularity, aspect ratio, roundness, and solidity. They are represented by all of these lovely formulas, but the main one that people use is circularity. And so we zoom right in and we look at number 9 and number 10. They're quite different in shape, and lo and behold, they have a different circularity. So that's how we can kind of filter out, right? So that's in that analyze particles. If we wanted to, we could say exclude things bigger or, you know, more round or more circular based on the numbers there. We kind of briefly did this before in the crystals image one, but we can do a plot profile. So what that is it's you draw a line and then you map the intensity along that line on a graph. So if we look at this red, green, and blue image, we can see that the red's definitely not where the blue is. We can see that the green's kind of everywhere, and that the red's definitely peaks at the perimeter. And that's kind of obvious from here. But this is a useful tool to show things like colocalization or that. One important thing is the actual gray values here don't matter, because if I just cranked up the blue laser, of course, the blue is going to be higher. It's more about the positional information. So like this is kind of in the middle, and there's where there's no red, there's where the blue is. And the other cool thing is graphs in FIJI have scale. So if I wanted to measure how wide the nucleus was, because I've drawn across it here, I could just draw a line here across there and hit M, and it'll tell me how long it is, because the scale's on the axis and that, of course. So it's a nice thing. So to put all of that together, now I'm going to use the blobs, but in the exercise coming up, you're going to have a nuclear picture. But we can kind of duplicate out that image just in case we stuff it up. So we're going to do a filter first. We're going to blur it. Go to blurry. And then we're going to go to our segmentation. So we're going to do a threshold. That works. So we can try one of the algorithms, apply it, Coolio, and then it's going to give us our new image. Now, what I want to do here is there's a couple of little things. So I might click on some of those little bits and measure them just to see how big they are. And I can look at the sizes and kind of begin to go, well, this is what I'm interested in. This is what I'm not interested in. Bring up the analyzed particles and not go 0 to infinity, but I can say if everything above 50 is what I'm interested in. I'm going to show a mask. So that's going to open a new window with just what it found. So I can visually look at that and go, yep, that makes sense. I've got them all stored here in my ROI manager. And now what I can do is I can go back to my raw image and show all of those overlays on them. And then I can select them all and measure it, bang, that's all my intensity of every nuclei in the image. So pretty quick to do it on that. Obviously that's postage stamp size image. That's an example one that works perfectly every time, but that's the kind of workflow that you can build up to do. So you might have cells and you wanted to see what's my transfection efficiency. So you can find all the cells and then just measure them all and go, which ones have a high green, GFP intensity or something. So that's one kind of workflow, but use your imagination that you can measure and count all different things. So we've got nuclei, and this is, I think, the last one of the day. Basically, repeat that workflow with the nuclei image and see if we can just measure the intensity of the nuclei. But in reality, you'd probably have like some marker that like translocated to the nucleus, and you would measure those ROIs that you generate and see which cells were there or something. But hopefully it makes sense, and